Hi, I'm Dr. Amy Turner, and welcome to Protein Tech's podcast, the platform where we talk to life science leaders and get to grips with the big questions of the day that help us to read the book of life. Our guest today is Dr. Layla Hosseini-Gerami, Chief Data Science Officer, or CDSO, and co-founder of Ignota Labs. Layla is a leading expert in developing machine learning models with a keen interest in computational predictions of drug mode of toxicity. In 2022, she was awarded her PhD in molecular informatics from the University of Cambridge. Her research focused on developing methods to better understand the mechanism of action of drugs using systems biology approaches and chemical analysis. The ultimate hope was to generate protein-protein interaction networks as predictors of compound-induced gene expression changes. Since then, she has co-founded and is the CDSO, or Chief Data Science Officer, for Ignota Labs, where she is responsible for the technical development of cutting-edge AI solutions for predicting, understanding, and mitigating toxicity issues in drug discovery. Her goal is to leverage AI not just to understand, but also to prevent adverse drug reactions and ensure safer therapeutic options for patients. Beyond her scientific career, she enjoys gardening, cooking, and listening to podcasts. So welcome to Protein Tech's podcast today.
Thank you for having me.
Let's get started. So in layman's terms, what is AI, specifically machine learning, and how can we use it in the context of drug discovery and mode of action?
Yeah, that's a really good question, because I think often AI and machine learning are kind of used interchangeably, and it's a bit confusing of what those actual terms mean. So you can think of artificial intelligence or AI as the kind of overarching term. In general, it just refers to machines that can perform activities that traditionally humans would do. AI can be found basically everywhere. I mean, I think a lot of us now have things like Siri or Alexa, and that's an example of artificial intelligence. It's kind of like an artificial human that's there and talking to you and doing tasks that you tell it to. Machine learning is then a subset of artificial intelligence, which is really focused on the idea that we can use data, or specifically algorithms can use data to kind of learn and understand patterns within the data to be able to predict things in the future. So in the context of drug discovery and kind of understanding what drugs do, we can apply machine learning to take all the kind of historical data about different drugs, what conditions they treat, different toxicities that they might show. And then the machine learning can then learn what is it about these drugs, which is leading to whatever effects that you're interested in, and then apply it to a new drug where we don't know what it does, and then understand based on that kind of historical data what is happening. So that's the kind of, in a nutshell, what all of that means.
Amazing. And how did you get into this? Where did it all begin?
So it actually started during a placement that I did during my undergrad degree. So I did chemistry as my undergraduate. I did an integrated master's, so a BSc and an MChem. And the program that I was doing at Leeds had a year in industry option. And I was really keen to do that from when I applied, just to have that kind of industry experience. I thought it would make me kind of stand out amongst the crowd for my degree. But back then, I mean, I think things have changed now, but back then there weren't any computational models. There was no kind of AI for chemistry module or anything like that. I actually didn't do a computational module until my final year, my master's year. And I was starting to kind of realize throughout the course of my degree that the lab wasn't really the place that I wanted to be. And that was something that was quite challenging, I would say, because what got me into chemistry in the first place was doing experiments and things like that. I found that really fun. Yeah. But it's a whole different ballgame when you're doing actually at university, and it's not just like a little 30-minute experiment in your science class at school. So I was really kind of struggling and really thinking, what can I do that is still related to chemistry, because I do enjoy chemistry, and I was really interested in drug discovery, but it just doesn't involve me being in the lab. And that's when I found a really good opportunity to do a year in industry for a drug discovery software company. So they develop software that pharma companies use, but as well as kind of other industries like agrochemical, cosmetics, etc. And within that company, they have different machine learning models to predict different properties of the compound. So things like solubility, for example, is a very important property to understand about your drug. So I got the opportunity to do my year of industry there, and I was building machine learning models to predict different drug properties. And I just absolutely fell in love with it. I hadn't done anything like that before. And I went in thinking, oh no, like, how is this going to go? But then quickly realized that it was something that I liked and that I was actually good at, which is a big contrast to the lab stuff.
Definitely. So from doing your undergrad in Leeds, where did you go from there? You've obviously got your year in industry. You've figured out that computational chemistry is something that you love. What did you do after that?
So during my year in industry, our CEO he's absolutely great. And he's actually on our advisory board now. But he was kind of talking to me about the placement and kind of saying, well, you seem to have really taken to this. Like, do you think this is something you want to do as a career? And I was like, yeah, it's great. I would love to do this every day. And he kind of turned around and said the dreaded words of, okay, well, you're going to need to go and do a PhD, because to have a career in this field, you tend to want people that have done a PhD in it, especially because it wasn't really something that was being taught during undergrad at the time. So I had quite a big decision to make at that point, but I knew that I really wanted to continue down this path. So what I actually did was I reached out to some professors. So the year in industry was in Cambridge. So I was reaching out to some professors in Cambridge. I found a group, the Bender group, and then I decided to do a kind of 6-week internship the summer after I finished my year in industry, before I then went back to Leeds to do my master's. And I got to see kind of firsthand the kind of research that was going on, kind of getting stuck in myself, even presented at a group meeting. And then kind of started to see, okay, well, I think I could do a PhD. It seemed like a very daunting thing at first, but then actually getting the chance to go and spend some time with the group, I think that was definitely a really good decision that I made, just to kind of try before I buy, so to speak. After that, the 6 weeks, I went back to Leeds. I did my master's and I made sure it was like computationally focused. It was looking at molecular dynamics simulations, not entirely related to what I do now, but I just knew, I do not want to go back to the lab. Like, please, I've spent a year away. Do not send me back. And then while I was doing my master's, I was then decided to apply to do a PhD in the group that I did my internship at. Cool. And then ended up getting it. And then that's what I went on to do next.
Amazing. So talk to me about what your project was in the Bender group at Cambridge.
Yeah, so my thesis title was, I can't remember the exact terminology. It's a very wordy, it's a very wordy title, but it was something to the effect of kind of using heterogeneous data types to better understand drug mode of action. So can we kind of look at different types of chemical and biological data to really get a good sense of what a drug is doing in the body to lead to its beneficial effect? Because a lot of the time drugs will get approved, for example, to treat cancer, cure a headache, whatever it might be. They kind of know that it works in like phenotypically, like they know, that it's shrinking the tumor, it's curing the headache. But what is often not very well understood is how it's doing it. So that was kind of my project, my task, my mission, so to speak, was to really kind of come up with some novel ways of using and combining data to get a really good picture of what the drug is actually doing.
Cool. I mean, that's crazy. I didn't really know that if you have a look at a drug and it's on market, everybody's using it. For example, paracetamol. Everybody uses this probably every week, let's be fair. And if the drug mode of action isn't known, I find that wild that it's able to even be approved. But I guess that's where healthcare has kind of evolved over the last few years. So I guess on a day-to-day level for your PhD, what were the methods that you were using? What were the methods that you were developing with your computational skills?
So I was using kind of 2 types of data. So the first was the one that I was familiar with from my year in industry, which was chemical structure data. So kind of taking data sets where we have a molecular target, for example, and then a kind of list of different compounds and their chemical structures and the extent to which those compounds engage that receptor. So the kind of first part of the work was really focusing on those types of machine learning models where we would take that data, train a model, and then we could say for a new compound, can we predict whether or not it's hitting this particular receptor? But that was very much kind of on the target level. So it's very interesting and important as well to understand what target your drug is hitting. But what you then don't get from that is then what happens next. So we can predict based on a chemical structure that this drug is going to bind to this protein. But depending on, where in the body that is, how much of it they're taking, kind of other the complexities of biology mean that it's hard to kind of guess what happens next.
Not a binary equation.
Yeah, exactly. So then I was combining that with gene expression data, because with gene expression data, it's a snapshot of the cellular response to a drug. You get some cells, you treat them with a drug, and then you see which genes have become over or underexpressed in response to that compound. And if you think of the kind of systems biology approach where you can say that, a drug will bind to a receptor, this will set off some kind of signaling cascade within the cell where the proteins are kind of talking to each other and transmitting messages until you eventually get down to a type of protein called an effector. So in this case, a transcription factor, which controls the kind of extent of transcription for different genes, and then therefore kind of controls gene expression. You can kind of see it as these different layers. And we can predict with the chemical structure the top of that layer, that molecular target, and what's kind of, I guess, starting the whole thing. And then with gene expression data, we can measure that the bottom of that, bottom of all those layers, which is, what's the response. So what I was working on was how do we connect the two together to get what's in between and what signaling changes are happening to lead to those changes in gene expression, given that a drug is hitting a certain receptor. And then we can understand things like what pathways are being hit, if there's resistance, for example, in certain patients, is there any other route in this pathway that we can take to be having the same effect? So that's what my work was really focused on, was combining cheminformatics and bioinformatics approaches together to really get that comprehensive view of what the drug is doing within the cell.
Amazing. That sounds so interesting and so applicable to a whole host of research areas, not even just in drug discovery or sort of drug mode of action. Because I think even could you apply this to how a hormone works, for example, and how like actual genuine native biological context things are happening? Is that something that's possible with the machine models that you were developing as well?
Yeah, absolutely. Yeah. So you might not always have, a small molecule engaging a receptor, but if you have that bottom layer of what's happening downstream, it's an approach that's called causal reasoning, essentially, that you use to kind of reason, if this happened, then what was the causal chain that led to that?
Yeah.
So it is quite applicable and different. Yeah, not only small molecules, but also other types of molecule.
Cool. And obviously you're coming at it from a computational aspect of you're getting all of this data and you're trying to figure out how you can analyze it in a useful way. Where do you get your data from in the first place?
So we got our data from a mixture of places. There are lots of publicly available repositories. So one resource is called Chembl, CHEMBL. And this is probably the best resource, I think, for cheminformatics out there, because it's a manually curated from literature and also from some high throughput screens, where you have for each of these receptors or targets a whole host of compounds and their activity levels. So this is like quite a good resource, which we use quite often. It obviously has its challenges, when you're taking data from, potentially different labs, different assays, different readouts, sometimes errors in curation and things like that. So it's definitely not perfect, but it's a really useful resource, a very unique resource that I think has been really important for our field.
And that's more the chemical aspect, so that top level data. Gene expression wise, was that also available on Chembl, or is that something that you take from sort of the Protein Atlas or the Human Genome Project? What sort of data sources do you use for that aspect as well?
Yeah, so during my PhD, I used a data set called LINCS, LINCS. And this is a very large data set of gene expression profiles across different types of cell lines for, I think, hundreds and thousands of different drugs, different compounds. So they very systematically profiled all these drugs in different cell lines at different time points and different doses, and made that data available for public use. So that was definitely also a really, really useful resource because it's a lot of information, a lot of data. Again, it has its issues. The reason why they could kind of gather so much of this data was because they were using something called the L1000 assay, which essentially means they were only measuring the activities of 1,000 genes and then inferring the rest of the transcriptome from that, which meant that they could do this very rapidly because only measuring a small number of genes. But then you have to think, well, there's the machine learning that extrapolates from those 1,000 genes to whatever, 10,000, 12,000, however many. And then if you're doing machine learning on top of the machine learning, then it can start to get a bit ropey.
Yeah, a bit biasy.
Yes, exactly.
Okay, so your PhD sounds incredible. I think that there's a lot of, like I said, a lot of applications of the sorts of models that you were building. After your PhD, what did you want to do? What was your sort of journey, career goals?
Yeah, so while I was doing my PhD, I got involved in an initiative called Downing Enterprise. So I was at Downing College when I was at Cambridge, and we had, unfortunately it's not running anymore, but we had a annual competition where students with, business ideas that, maybe their research, they realize that they commercialize it, would come and pitch in front of a panel of judges, which were basically rich alumni from Downing College, and could compete to win seed funding for their business. And I was kind of looking for a job during my PhD, and I saw that this was, working and kind of doing the operations for that was out there. Decided to go for it, and then just became very inspired, kind of seeing these students come in with their amazing ideas, and kind of also hearing, on the other side, you hear the student pitch, and then they leave, and then you hear the judges kind of talk about, what do we think about their business model, and their ability to generate revenue, and kind of using all these businessy terms I had never really heard before. But that definitely set a spark in me of it'd be really cool to have my own company one day. But I think I was kind of feeling imposter syndrome. I know everyone has imposter syndrome to some extent, but I was kind of telling myself, well, I can't do that. I can't start my own company now. I don't know anything. Maybe I'll go and work out in a big company for a few years, get some experience, make some connections, and then maybe I'll feel ready to do my own thing. So what had happened was I was writing up, it was the beginning of 2022. And while I was writing up, I was kind of looking for jobs, seeing what was out there. And what most people in my research group did was go off and work for Big Pharma. There is a big demand in Big Pharma for these kind of computational skills. And a lot of us were actually sponsored by pharma companies as well in terms of our PhDs. So I kind of thought, well, I'll go off and work for AstraZeneca for a few years, and then let's see what happens there. But what had happened was I was starting to think, is this really something that's going to make me happy and that I'm really going to enjoy? Mainly because during my PhD, I was very independent. My PI wouldn't say you should do this. He would say, well, what are you going to do next? And it was really up to me to kind of decide all of that. And, I didn't have a boss breathing down my neck. Because as I said, I was a very independent researcher, and I really enjoyed that. And I thought, well, if I go and work for another company, especially a big company, I'm just a very tiny cog in that massive machine. And, it might take me a few years to maybe climb up the ranks to get to the point where I'm the leader of, for example, the data science team, and I can actually help shape what research is going on rather than kind of being told what to do. So I think it was that kind of autonomy that I was craving. So as well as looking at big companies, I was also looking at, small companies, are there any jobs happening, etc. So I'd had a kind of a few interviews. It was still quite early days in my job hunt. And then one day I got a message on LinkedIn from someone I'd never heard of before in my life.
LinkedIn.
I know. It's so good. Yeah, just someone I'd never heard of before, just a complete stranger saying, hey, I'm starting a company. It's about using machine learning to predict drug toxicity. We need a machine learning lead. Are you interested? And the first thing that happened was imposter syndrome kicked in. And I was like, oh, I think you've got the wrong person. You know, I'm doing my PhD, you know, I haven't finished it yet, you know. And then
from the outside though, from me listening, I'm going, this person has found the exact right person. They're doing a PhD at Cambridge in machine learning about drug toxicity.
And that's in fact how he found me.
Right.
Yeah. So he had kind of, so it was my my now co-founder, Jordan. He's been in drug discovery for a decade. So he's worked across big pharma, CROs or contract research organizations, and also AI drug discovery companies. So he's had quite a broad view of the industry. And my other co-founder, Sam, is a complete business whiz, went to INSEAD to do his MBA, has worked with the AlphaFold team. So he has like experience in translating like the tech to the healthcare and kind of bridging that gap. And they were like, well, that's great and all, but we need a machine learning person. So Jordan, my co-founder, the scientist, had he kind of knew that my group was an AI for drug discovery group, and had kind of figured out that I was finishing soon. So that's why he reached out to me. But at first I was like, really? Are you sure? Like, I'm fresh out of my PhD. How can I do any of this? So at that point, again, like I had a big decision to make.
Yeah.
So I went and I actually spoke to my PI. So my PI, Andreas Bender, he's actually started a few companies himself. And what he basically said to me was, just go for it, because even if it goes wrong, even if the company lasts 6 months, a year, whatever, you know, you're just starting out in your career and it will only ever be like a good experience and kind of building new skills for you. So he said, I really don't think you need to worry about this being a big risk-taking thing, because ultimately, if the worst happens, you're not going to be worse off. Like, you can just go and find another job.
Yeah, very good advice.
Yeah, exactly. So he was kind of basically telling me like, just go for it, because what's the worst that can happen?
Nothing to lose.
Exactly.
Quite a lot to gain.
Exactly. Yeah. So I think that then really pushed me in being like, yeah, I think I can do this. So, you know, I had a couple of video calls with Sam and Jordan. They took me through their pitch. And then we met in person, met up in Cambridge for the first time. And then it was evenings and weekends of investor calls and prototypes and things like that, until we managed to close our first funding round. So yeah, that was definitely intense, kind of starting a company and finishing a PhD at the same time.
Mm-hmm. Oh yeah.
Yeah. But I wouldn't change a thing. I wouldn't go back and change it.
So what is the company pitch? Talk to me about Ignota Labs.
Yeah, absolutely. So drug discovery is very hard. Lots of things can go wrong. And something that goes wrong quite often, it's actually up to 56% of the reasons why a drug fails is because of toxicity issues or safety issues. So the drug might go into an animal, and the animal shows some toxic side effects, or it might even go into a phase 1 trial, kind of the first time it goes into a human being. Patient might have chest pains, raised liver enzymes, what have you. And as I said, this is extremely common. The issue with this is, even though, you know, these animal trials, these human trials, they tell you that something has gone wrong, what they don't tell you is how to fix it and what to do next. So the consequence of that is that all of these drugs just get thrown away, essentially, left on the shelf to gather dust. And
these are drugs that have been taken, 10 years to get to the point that they're at.
Exactly. Yeah.
Right.
Yeah. Because it's such a long, hard, time consuming, expensive process. But ultimately, there is so much uncertainty once you get to that point of the drug has shown toxicity, because at that point, the question is like, well, okay, where do we go from there? And the easiest thing to do at that point is to say, we give up, we try something new. And this amounts to about $400 billion of value being lost every year.
Every year?
Every
wow,
year.
Okay.
Yeah. So at Ignota Labs, we want to give new life to these drugs. So taking these drugs, really understanding what went wrong and answering that question of what can we do next and how can we fix it. So we take drugs that have safety problems, understand what went wrong, fix them, turn them around, and then we put them through a preclinical study, so back into an animal trial, and then into a phase 1 clinical trial. So the first time it goes into humans, and then at that point, we sell them the drug back to the pharma industry. So we're kind of flipping drugs, like instead of flipping houses, we're flipping drugs. But it really means that we can give new hope to patients that are still waiting for these drugs to come and treat their disease. And instead of these drugs being left on the shelf, we can kind of give them a new life.
Amazing. I think that's quite a niche area, but something that could have a massive impact in the current drug discovery pipelines, the preclinical, post-clinical, all the way through from sort of the start of a research project all the way through to actually put it into a patient. Yeah. In my mind, it's like 10 years of research, people at the lab bench, PhD students, masters, postdocs, a PI's full career, finding this one shape of a jigsaw piece, putting it into a mouse or a human and realizing that it's wrong and that jigsaw piece gets put in the bin, but all that needs to be done is the little bit, the little circular bit of that jigsaw piece just needs to be moved around a corner.
Yeah.
And then it could be fundamentally impactful and saving lives. Is that sort of something that they're using the jigsaw piece of changing it around a little bit?
Yeah.
And it can repurpose that.
That's a great analogy. Yeah. So we really try and make very small changes. And I think the benefit of what we're doing is these drugs that we're looking at is they're clinical candidates. Like they're good drugs. Apart from their toxicity, everything about them is really good. Like metabolic stability, actual efficacy, so how well it's able to do the thing that it should be doing. So it's really just that one piece of the puzzle, to go back to the jigsaw analogy, that we're trying to fix, which is a really good thing for us because, as I said, drug discovery is so hard. And even to get to that point where you have something that's good, but it's just toxic, takes so long. So we're kind of really at an advantage here because we're starting out with something that's already pretty good. But then that also means we don't want to change it too much because we don't want to then start messing with the bits of it that are good. So what we do is at Ignota Labs is all of our machine learning is explainable. So we don't just get a prediction and with no context behind it. It's like the drug is doing this thing and that's it. What we do get is we get a kind of molecular map. So if you think about a chemical structure, you've got your different atoms that are all joined up by bonds. We can kind of highlight which parts of that structure does the machine learning think is driving that unwanted interaction.
Okay.
So then we can use that information to inform what changes need to be made. And it might be the case that whatever part of the molecule is driving that unintended interaction is actually also driving the intended interaction with the actual target that you want it to bind to.
Could you then think about dosing in that context?
Yeah.
So obviously looking at it from a pharmacological perspective, whilst the drug might be having its intended purpose, obviously that purpose could have a toxic side effect as well. But if it's just need to find the balance of how much you need to give to a patient, is that something that your system could also inform on?
So it's actually something that's kind of already used as a strategy within drug discovery, but it often doesn't work.
Right.
So you'll see a lot of, as I said, 56% of drugs fail due to safety, but then, some drugs will fail due to efficacy reasons. So they haven't met the endpoint that they need to meet. But oftentimes this is kind of a safety issue in disguise because what's actually happening is they can't give as much of the drug as they would like to the patients that they need to, for example, shrink their tumor without causing that side effect because what is called a therapeutic index. The therapeutic index is so small that the amount that you need to give to someone to do the good thing is also going to cause the bad thing to happen. So what we like to do is really try and increase that safety margin. So we want the target, the drug to bind to the intended target at say like sub-nanomolar concentrations. And then ideally not hitting any unintended off targets, but if it does at a much higher concentration, like you'd really need to give a lot more than you need to cause that side effect.
And are you developing the machine learning models in-house, or is this something that you're repurposing from another place? Or is that something that's your role?
Yeah. So that's my role. Yeah, exactly. So my role is to kind of lead the research and development of our tech stack, and for us to kind of really, and as I said, it's quite similar to the work during my PhD, and it is really kind of building upon that. And for that, we need quite a few different models, one to look at the biology, one to look at the chemistry. We've also got a model for like, actually, how do we find these drugs that are failing, and kind of doing a almost like a sales pipeline, powered by AI as well.
I was going to ask about that. How do you find these drugs that are failing? Is it readily available?
It is. So there's lots of information out there if you know where to look. So where we get this information from is things like there's websites called Fierce Biotech and TLDR Biotech. These are two kind of news sources that report on drug failures, particularly within biotech, where these are small companies that, if they have a failure, it's not as simple as, oh, I'll move on to the next one. It's, oh God, this is our whole company and it's all burning. And so those places are good places to get that information. And then also clinicaltrials.gov,
right,
where you can see kind of where clinical trials have been withdrawn or suspended, or like maybe they started like 5 years ago and there's no update. So you're thinking there's something fishy here, something's gone wrong here. So that's the kind of top of the funnel. And then what we've developed in-house is a tool that's essentially like an AI researcher.
Cool.
So instead of having to employ, hundreds of scientists to go and do research on the drug and what went wrong and what it's doing and things like that, we use AI to do that instead. So what the agent will do is it will find out firstly about the drug, why it failed. There might be a paper where they have, the company have reported on the clinical trial outcome, and they might say patients experience serious adverse events, and kind of list them out so we know what's going wrong. But it will also kind of find out what's the target, what's that drug trying to hit, if it's known. As I said, it's not always known. What indication is that drug being used for? What's the market size for that particular indication? Can the drug actually be used in a different indication because whatever target it's hitting is actually relevant for a different disease? And then also things like finding the original patent from where the drug came from. And then you can extract chemical structures from that patent and already kind of start running it through our tech stack to start generating some hypotheses. And then as well as all of that, we have a kind of fact checker, because we know that the AI researcher is based on a large language model. And I'm sure we've all heard of the hallucination, right? Like the language model just making stuff up.
Yeah.
So we fact check all of our LLM, large language model information, by cross-referencing with resort like the primary sources like papers, again, information from these kinds of websites, and essentially asking, is what's written here and what's been found by the researcher actually substantiated by any actual source? And then we get a kind of fact check score, where often if something isn't necessarily available to find online, the language model has more of a tendency to hallucinate and make something up because the information just isn't out there. And they're very bad at saying, I don't know. Right. Even if you prompt it and say, please, if you don't know, just tell me you do know, not know, stop making stuff up. It will still make stuff up. So then that allows us to see kind of where are we less confident in what it's come out with, because there is nothing that's actually like corroborating what the AI has come out with. So what we get from this whole process is a report that a scientist can read that's broken down into lovely sections with a nice executive summary, a fact check score and sources. And essentially means that we can prioritize what lead do we want to follow, which looks like the most interesting opportunity, maybe in a very interesting disease area, or maybe the hypothesis we've come up with is very strong. And then we then start contacting these companies and basically say, we heard there's a problem and we can help.
We saw it. Yeah. You were trying to hide, but we found it.
Yeah.
So what kind of disease areas do you prioritize? Is it cancer? Is it diabetes? What is the mission of Ignota?
So for us at Ignota, what we definitely want to focus on, at least while we're still proving ourselves as a company that's able to do these things, is focusing on disease areas where we know there's a market for it. Like we know that we'll be able to sell the drug on, because we're not in a position, at least at the moment, to continue the drug through all 3 phases of clinical trials. We kind of get to phase 1. At that point, we've shown it's safe, whatever hurdle it fell down on, we've overcome it, and then sell that on. So we need to make sure that they're areas that are generally of interest to other companies. And we're also avoiding neuro, just because brains are very complicated, and the blood-brain barrier and neurotoxin, all of that, we're kind of saying, right, we'll stay away from that. And also very complex endpoints, because while we're doing this phase 1 trial, it's primarily to see, is the drug safe? Can a human being take it and not have a heart attack?
Yeah.
But also, it would be good to have some kind of signal, is like, is the drug working? And indications like cancer, for example, or obesity, it's a very easy thing to measure. It's very simple. Has the tumor shrunk? Has the patient lost weight? But when you're thinking of indications, maybe like Alzheimer's, for example, it's a lot more complex. So we're sticking to, disease areas with a kind of clear endpoint and that have a market to sell to. And that's our kind of overarching, but that's not to say that we won't pick up a opportunity that might be very interesting, even if it doesn't necessarily fit in that box. It's primarily oncology, cardiometabolic disease are our main kind of areas.
So Ignota Labs is obviously a very new company. It's something that you've set up from scratch. How does it differ to the current companies that are in the machine learning and drug toxicity space at the moment?
That's a really good question, because that's also something that investors would ask us a lot. There's definitely been a big explosion in AI for drug discovery. And it's often hard to kind of tell what is different about the different companies. But I think what I would say is we're taking a different approach to a lot of AI drug discovery companies out there who are trying to do the whole thing end to end. They have a really cool piece of tech that's doing something really interesting, like maybe finding novel targets, for example. But ultimately, as I've been saying, drug discovery is extremely tough, and that one tool isn't going to help you throughout the whole process, right? In our case, we're focusing on a very narrow slice of the drug discovery journey. We're taking things that have come out, they're already a clinical candidate, they've come out of a trial. We're fixing it and then putting it straight back through the trial. Like, it's really only like a 3 to 5 year window, as opposed to like the 10-year plus journey. And we're really focusing on the area where our technology has its strength, which is understanding toxicity. And that's what we're focused on. And I think in the future, we'll kind of move towards a world where we'll have different AI drug discovery companies who have their own particular niche that work together, maybe, and there's like going to be a marketplace or something where different companies can come together and we can say, hey, we've just found a drug and we've found its toxicity mechanism and we've turned it around. Now we really need someone who has a machine learning model to understand this particular type of cancer that we're trying to treat, to really help us make sure that we're not losing its efficacy when we change its chemical structure. And then kind of working together to help each other, because, as I say, it's such a long and difficult process that I think that's where maybe we haven't seen the big impact that we thought we would see from AI drug discovery thus far, and actually getting drugs into the clinic and into patients. Like, that part really isn't happening.
It's not a one-size-fits-all kind of learning model.
Yeah, exactly.
You definitely need the different modalities to work together. That sounds amazing. I think Ignota Labs has got a successful future in your hands.
Thank
you. Good. How do you foresee your company growing? I think at the moment you've had 2 years. You started 2 years ago, and from the outset, it looks like you've had tremendous success. You're the only company that I know at the moment that is doing this sort of work. And it's obviously, if it is going to work, it's going to have amazing impact of the whole healthcare and drug discovery areas. Where do you see it going in the next 5 years, and what areas do you want to grow into?
Yeah, so I would say the last couple of years has really been about developing the tech stack and putting the building blocks in place to be able to do this drug turnaround. We're now at the point where our kind of next horizon is actually doing the drug turnaround. So we've in-licensed 2 drugs so far that we're going to be developing and working on in-house, and hopefully purchasing a few more. And in 5 years' time, we want to have successfully turned around a drug, successfully taken a drug that had issues, fixed it, and proven that it's safe in an animal, ideally in a human, but if not, then in an animal, just so that we can kind of prove that this is something that we can do and to keep going with it.
Do you think in terms of the amount of people that you're going to have to collaborate with, is it going to be collaborative? Are you going to work with other companies to get this into get your drug into different trials, or are you going to have in-house scientists that you're going to have your own labs that people can work with at the bench? Are you going to be able to go from your office working on your machine learning models and then pop downstairs and go, hey, can you just put this drug in there? What's it going to look like?
I think it would be great in the long term to have our own labs. But for now, we're just solely computational based, and we're using contract research organizations to carry out the wet lab work. And the reason for that is, as I say, we've got the two drugs that we have in two different indications. One's for cancer, one's for cardiometabolic disease. The kind of we'll be running very different experiments as new drugs come in. So instead of having to have like a very well-equipped lab that's able to do all these kinds of different things, it's easy for us to just go to CROs who are very specialized and do kind of small package of work with them. But we are hiring in project leaders for our drug discovery programs. So people who have expertise in medicinal chemistry and biology in the context of drug discovery to help oversee the projects, liaise with the contract research organizations, and then also work with the tech team who are working on the tech stack as well. So yeah, for now it's going to be kind of outsourced. But as I say, like maybe in the future we'll have our own labs, who knows?
When you have world domination, you can have everything.
Exactly.
So going back to, I guess, the wet lab stuff as well. So at the moment you're working on models that are taking from data that's coming from lots of different sources that might perhaps not be standardized. How does that challenge you in your development of these models? And how do you think that might change in the future?
Yeah, that's a really good question, because I think there's two sides to the coin in that, okay, there's lots of data out there for especially this kind of compound target interaction data. But then the issue is that it's very unclean and it needs to be refined. So it's kind of, they say like data's like oil, there might be an abundance of it, but you need to refine it and make it into something that's actually usable. And the problem is that when you start refining it, it just becomes smaller and smaller and smaller and smaller until you end up with a very tiny data set.
Yeah.
And if you think about, the machine, it needs enough samples to actually learn patterns. So if you give it like 3 drugs, like how is it going to extrapolate to anything? So it's a real challenge because you want to have enough data for the machine to learn. But what you don't want is to add a bunch of noise with your data where it's kind of trying to predict 10 different things at once.
Yeah.
And I think in the future, this is going to change because I think we're starting to realize that, machine learning is a thing and it's not going to go away. And I think when it comes to wet lab experiments, things like ensuring that the metadata annotation is thorough and complete is going to be very important. And kind of you almost need to assume if you're in the lab that your data, whatever comes out of it is going to be used in a machine learning model.
Yeah.
And I think it's really about kind of scientists and machine learning practitioners to kind of work together and to hopefully build a world where we have data that's, machine learning ready that can be used to inform us in the future, because I think that's an amazing thing to do. Like all these experiments that have been run, the fact that we can use them to develop new drugs and to keep improving and keep learning, I think is really cool.
I think you're an excellent example of how it needs to be more integrated into kind of education at an early stage. So obviously you told us about your undergrad and not really, learning about chemistry, but then thinking, I'm not really sure whether I want to be in the lab at the bench doing my chemistry. I want to learn about computational biology or computational chemistry, and you thinking, this is actually something that I want to do, and now turning that round after doing your PhD into a company idea at the moment that could be amazingly successful. But had you have had the computational multidisciplinary element of your current career at that early stage, I feel like there'd be such more people doing this sort of thing. And I think we are definitely going into that kind of multimodal level of data analysis. And I think in the future, it is going to be a case of, I'm going to do a Western blot in the morning, and then I'm going to analyze that data, and somehow that's going to train a model in my afternoon, and it's going to be one person who knows all of this and have kind of such a complete and holistic idea of what the life science research space should look like. I think that's really cool.
Yeah, I definitely agree. And it's something that I've noticed, like I've been asked to deliver talks to toxicologists and wet lab scientists about AI and machine learning so that they can learn and understand. So for example, I did a talk at the ECHA European Chemicals Agency in Finland. And what they were saying was like, we get obviously a lot of regulatory packages coming in and saying, you know, this is our chemical and these are the different properties of it, et cetera, et cetera. This is the potential hazards related to it that are actually coming from machine learning models.
Okay.
And they don't quite understand, you know, how was this machine learning model built? Or even like, what questions do I need to ask about this machine learning model, right? So I went in to kind of tell them like, this is how these people are building their machine learning models. Like, it's actually quite simple if you kind of break it down and kind of show what's actually happening under the hood. And then kind of saying, well, when you get these models, you need to ask these questions, like what data was it trained on? What types of chemicals was the model trained on? Because if you have a bunch of chemicals that look very similar to each other in your training set, so whatever the data set that the machine is kind of learning from and learning these patterns from, if you then were to take a drug that looks completely different from anything the machine learning model has ever seen, you're not going to be able to trust that prediction just because I say, you know, the machine has not seen it before. An analogy that I like to say is like, pretend that we're building a machine learning model to predict house prices. And we're in Manchester and we're going around and we're going to different houses and we're measuring the square footage, we're measuring the distance to the closest school, you know, things like that. Kind of different features or attributes of a house that might then kind of lend itself to whatever price it's going for. And we do all of this, we get all our data and we, you know, find the patterns and we say, okay, great. Then someone comes along with some information about a house that they saw in London. You might have very similar square footage, same distance to a house or a pub or whatever kind of metric you want to use to say that, you know, it causes change in house prices. But that price is going to be extremely different. Mm-hmm. You know, so that's similar to...
It'd be lovely if it was the same though, wouldn't it?
I know, it would be great. But unfortunately, that's not the case. And it's the same with the chemicals. You know, they might look a certain way and then something looks very different. The machine learning model isn't reliable. Yeah. So you need to be able to understand like, where does it work? Where does it not work?
Yeah.
And I think there's going to be more of that of, you know, wet lab scientists really wanting to learn and understand about what machine learning is. Because as I say, like, you can't escape it anymore.
No, it's everywhere.
Yes, exactly. Yeah. And even if you never use it yourself, just to be aware of how it works and what it means, so that if ever you do use a machine learning model, someone says to you, I'm making a decision off the back of something that a machine learning model has said, you at least know kind of what questions you need to ask to see if you can really trust the machine.
It's definitely going to be a tool that's going to be a common tool in the toolkit that anybody's going to be using, not just in the life sciences, but elsewhere as well. Going off the idea of having machine learning as a tool in our toolkit of life science research, I keep seeing this idea of explainable AI, and obviously AI is becoming more accessible on the sort of layperson's feed, I guess. Do you think it's going to become something that is absolutely integrated into literally everything that we're doing, and especially on the research side of things?
I think so. I think we're going to start learning over the next few years where it can best be used. I think you describing it as a tool in the toolkit is a very good way of putting it, because I think there's a lot of fear around AI replacing us. Mm-hmm. But I think the idea is more about efficiency and automation where it's possible, but then also recognizing that the human being is still needed in that equation. Mm-hmm. You know, for example, if you had a team of data scientists that wanted to build a model to predict if a drug's going to bind to a target, they might go on Campbell and download all the data there is and chuck it in a machine learning model and say, here you go, we've got an amazing model. What they don't have is the biologist who is saying which assays have these data points come from. Maybe we have some that you've taken a single protein that you've purified and you're just seeing, does the drug physically bind to the protein? Or there might be other assays where the protein is in its cellular environment, and perhaps the drug, which would have bound in a single protein situation, can't get through the cell membrane and actually kind of permeate and bind that target in that scenario, in that context. And that's really important to be able to see like where that data has come from and really understand that. And that's why we need the scientists to be able to do that. And then also the chemists kind of coming in and saying, well, what's the diversity of your training set? You know, is it all very biased to one particular area of what we call chemical space, which is kind of all the potential chemicals that could ever exist, of which there are many. I think it's 10 to the 60.
Wow.
So a lot of these models are very biased because they're in a very tiny pocket of chemical space. And again, as I said, really important to understand that. So in that scenario, it's really about like the AI practitioner, the chemist, the biologist, kind of all working together and using that as an important tool, but also recognizing its limitations.
So you're working in quite a niche area of AI and obviously the drug toxicity space, if we're going to use that word. And then you also mentioned obviously you need humans, you need people, scientists from all different disciplines, the biologist, the chemist, the pharmacist. Could you foresee, obviously with the boom in AI, it's like you said, it's inescapable, it's happening and we have to keep up. Could you imagine in the next 20 years, I'm hopeful, I've said this before, but I'm hopeful that there's going to be sort of a digital human. And obviously I think you work with liver, like mainly liver toxicity. There is obviously models at the moment that can semi-digitalize what a liver could look like, and you add in compound A, what happens to the cells in this area, what would be the output of that? Do you foresee that if all of these different disciplines could work together and could understand AI and its utility, that you could build sort of this massive model of a human in all types of disease states?
Yeah, I think so. I mean, it's definitely a bold, not a bold claim, but something that will have a lot of impact, and that would be extremely impressive if it can be pulled off. But I don't see why not. I mean, we have lots of advances in so-called digital twins, as you said, of livers, of hearts, etc. So I don't think it's beyond the realm of possibility that we'll have a full digital twin of like an actual human being one day. And that will be really amazing because, when you have, patients, people suffering with illnesses that are going into clinical trials, they're kind of, hanging all their hope on this drug working. So I think when they actually end up getting sicker because it's causing a toxic side effect, I think that's such a real shame for these patients that it's kind of their last hope and it's actually making them worse. So I think if we could put these drugs into a kind of digital twin where we could actually not only have like a digital twin or a replica of the kind of average human, but kind of specific humans, like can I take Amy and make a digital twin of her who would react very differently potentially to a drug compared to me. Yeah. So, digital Amy and digital Layla, let's simulate and see what the differences are. I think
it's a big game of The Sims, really,
isn't it? That's what we're heading towards. Yeah.
That would be, I mean, it's just another level to precision medicine there, isn't it?
Yeah, absolutely.
Very cool. I think you're working in such an amazing space. So I'm going to take us all the way back to PhD you. What would be the advice that you would give to yourself and anybody else who from PhD is thinking, I'm going to set up my own company? What steps would you take to better train yourself in that regard?
Yeah, so I think something that I kind of wish I'd done, which I'm still like toying on whether or not I want to do it, is actually get some kind of formal training in running a business. So something like an MBA. You can do them online now. So actually one of my colleagues got his MBA online. And I think that would have given like a lot more of a foundation as to like how to start a business and, deal with investors and kind of all the things that you don't really know until you do it. I mean, I've picked up a lot of it from just experience. But I think definitely when I first started, I felt completely out of my depth and I was like, I don't know any of this means. So yeah, even if you can't go and actually get an MBA, just kind of engaging with resources for teaching yourselves like the fundamentals of business, I think would be a great idea. And also just like not doubting yourself. I think when you think about starting a company, at least the first thing that came into my mind was like, yeah, well, 90% of them fail.
You're on the back foot straight from the off.
Yeah, exactly. And I think it's really easy to focus on that and to let that kind of be a reason not to go for it. But I think like especially when you're coming from your PhD, like as my PI said, like you're really in a situation where it will only ever be beneficial for you to go and do this. And you need to just kind of have faith in yourself and kind of just go for it if that's something that you find interesting. And also, for example, you might be the scientist who's come up with an amazing idea and wants to commercialize it. You know, it's hard doing it alone. And I'm very glad that I didn't have to do it alone. I had 2 co-founders and we all had like a very, very complementary skill set. Yeah. Like if you did a Venn diagram, the overlap would be quite small. Like we all bring our own expertise into the company. Yeah. And I know that when I was working for Downing Enterprise, we would have sometimes, you know, someone come in by themselves and just say like, hey, I have the kind of scientific idea, but I need someone who's like financially minded, etc. So kind of reaching out and finding other people that can join you on your journey. And a lot of universities do have kind of entrepreneurship, maybe societies or even like a department, for example. So engaging with those, any events that they put on, any talks, I would definitely also encourage.
What do you think are the transferable skills that you already had in your PhD, that you were already developing in your PhD, that have been useful during your time at Ignota Labs?
Problem solving, troubleshooting. So, you know, when you're doing your PhD, sometimes you've got, you know, a hypothesis, a research question, and you start working on it and you realize quite quickly that actually this isn't going to work, and you need to think of something else to do. And it happened, you know, a few times during my PhD. I'd say, right, I want to start researching this thing, and then, oh, actually that's not going to work. I need to pivot and do this other thing instead. And that's also very true of being in a startup, because you need to be very kind of reactive to things changing, like the market changing, for example. Or in our case, we actually started off using our technology at the beginning of drug discovery to predict whether a drug was going to show toxicity. But ultimately, that business model just wasn't working for us. So I think the fact that I was used to kind of being faced with something that wasn't working and actually saying, look, I need to fix it, instead of doing what a lot of companies and co-founders can do, which is be very stubborn and say like, no, I said I was going to do this. I'm not doing anything else. I don't care what you tell me. I'm going to keep going down this road to nowhere. And actually instead thinking, okay, what can we do next? Let's be light on our feet. Let's pivot. Let's try something new.
I always say that one of the biggest things that I learned during my PhD, other than the very specific research area that I did, was the ability to fail. The ability to just sort of go, this isn't working, and it's not personal. It's just, you know, it's just fact. This is not the route that I'm taking is not going to work. And where can I actually best utilize my time is not flogging a dead horse. It's going to be using my skill set or the things that I've learned during the failure of this and pivoting somewhere else. So I think it is such an important thing to not only recognize obviously for you, like the incredible machine learning skills that you had, but also the softer skills that people often forget when they're doing their PhD. So yeah, bringing that into a business, regardless of whether you've got an MBA or not, I think is something that is really important. So yeah, what in your journey, it's a novel journey. It's not something that you're the only CDSO that I know from PhD. Have you enjoyed it? Do you think you would have done anything different?
I really enjoyed it. I mean, don't get me wrong, there have been some very stressful times. I would say definitely the start of the company when I was writing up. And then also things like fundraising can be super stressful. But I think ultimately the kind of goal of the company and the impact that we're going to be having on patients and on the industry as a whole is what keeps me going throughout those moments of stress and, things not working, etc. So yeah, I definitely wouldn't change anything. If I could go back and speak to my PhD self, I'd be like, do it, send that message. Don't even bother talking to your PI, just do it, because it's been so much fun and it's just been amazing.
Layla, thank you so much for joining us today. I've had such a fun time recording this podcast. And I'll say it, if anybody wants to get in touch with you to learn more about machine learning and drug toxicity, or if they want to collaborate, please do get in touch with Layla.
Yeah, absolutely. And thank you so much for having me. I've had a great time.
Perfect. So to our listeners, if you've enjoyed this podcast, please like and subscribe to Protein Tech's podcast, where you can learn more about the big questions of the day that help us to read the book of life. Thank you for listening.